So we all rely on the ecosystem services provided by trees. And a key component in assessing these processes is understanding how trees are affected by factors like disease. Like us, plants rely on water and nutrients for survival. Carbon, hydrogen, and oxygen constitute the majority of a plant's dry weight, but plants also require a number of inorganic nutrients like nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. Nitrogen in particular is an essential component in plants. It's primarily absorbed from the soil in the form of ammonium and nitrate. It's essential for chlorophyll production, which is fundamental for photosynthesis, and it's also required for a number of plant cell components listed here. Interconnected water-conducting xylem tubes run throughout the plants. Water and nutrients enter plants through the roots, and then through a combination of the properties of water, structure of the xylem, and transpiration, the water and nutrients are drawn upwards to the leaves of the tree. This water column fails if air bubbles are inserted, which can be caused by a number of things. One possible cause is bacterial leaf scorch. Bacterial leaf scorch, or BLS, is caused by the bacterial pathogen Xylella fastidiosa. When large populations of this pathogen invade the xylem of a plant, the plant overproduces defense compounds, causing air bubbles to form that partially block the flow of water and nutrients to the leaves. This reduces the leaf tissue available for photosynthesis and also depletes starch reserves, resulting in the reddening of leaf edges, known as leaf scorching. The bacterium is vectored by insects that feed on xylem fluid, like leafhoppers and sharpshooters. And while the disease doesn't seem to spread very rapidly, there is also no cure. Symptoms appear late summer to early fall, and they vary by host, but the most common symptom is marginal discoloration, or leaf scorching, which you can see there. This is followed by a premature browning of the leaves, and affected leaves also drop early as well. And then gradual defoliation of the branches and a thinning of the crown. So, like I said before, decline is very gradual. The symptoms become worse over the span of 3 to 8 years, with more and more branches dying from defoliation, until finally the tree succumbs to the infection. And within populations of trees, the disease also seems to occur very randomly. Like you can see in that picture, the tree on the left is affected, but its neighbor is perfectly fine. Symptoms are easily confused with other diseases and abiotic stressors, so it's best to confirm with a genetic test. Xylella fastidiosa affects a very broad range of organisms. It causes decline and leaf scorch in shade tree hosts like elms, oaks, maples, and sycamores, but it also causes economic losses in many agriculturally important crops like grapevines, citrus, and coffee. Understanding how trees are affected by disease will improve our ability to diagnose plants in the future, and comparing the leaf traits between diseased and healthy trees will provide insight into the physiological responses of trees to decline. Therefore, my objectives were to test Mongolian oak branches within the Morton Arboretum collection for bacterial leaf scorch, compare leaf traits between diseased and healthy Mongolian oaks, and then also determine the impact of cardinal direction, age, and location on leaf traits. Because the pathogen blocks water and nutrients from reaching the leaves, I hypothesized that the moisture content for leaves from diseased Mongolian oaks would be lower than the leaves from healthy Mongolian oaks. Research has also shown that nitrogen is involved in the production of chemical compounds that protect plants against parasites and diseases, so I also predicted that the percentage of nitrogen would be lower for diseased Mongolian oaks. I also hypothesized that age would be positively correlated with all the leaf traits that I was looking at, percent nitrogen, percent carbon, moisture content, and leaf area, because older trees would most likely have a higher capability to store water and nutrients in the leaves. I sampled 11 open-grown Mongolian oaks in 4 different locations within the Arboretum. First, I measured the diameter of rest height and the height using PBH tape and a laser rangefinder. And then because decline is very gradual and portions of the tree can remain unaffected in the early stages, I collected second-year growth from the 4 cardinal directions for each tree. I extracted DNA from these twigs, and then to test them for presence of Xylella fastidiosa, I amplified the pathogen-specific DNA sequences using the polymerase chain reaction, or PCR. For my experiment, I was targeting a 766 base pair region of Xylella fastidiosa genome with the primer set RST31 and RST33. I ran my samples through a thermocycler, and then I loaded them into Agrostrel for 110 volts for 20 minutes. I also collected 20 sun leaves from each tree, 5 from each of the directions. The samples were immediately weighed, and then I photographed them with a ruler. I used image J to analyze leaf area. The samples were then oven dried for 4 days, and then I measured dry weight. And using the fresh weight and the dry weight, I was able to estimate the percentage of moisture content in the leaf samples. The dried samples were then ground and then wrapped in tiny tin boats, and then these tin samples were ran in an elemental analyzer, which told me the foliar nitrogen and carbon. Unfortunately, I ran into several roadblocks while working towards my results, especially with the genetic component. For my first few trials, I wasn't really getting anything, so I decided to take a step back and troubleshoot my methods to see what was going wrong. So the very first thing that I did was run a gel after extracting my DNA, just to make sure that there was actually DNA there, and I found that there was, and you can see. So then I moved on to the PCR component, and the first thing I did was test my PCR kit. So I borrowed different reagents from several of the other scientists here, and I ran tests using their reagents, and I quickly found that my Taq polymerase was not working. So I switched that out, but I still wasn't getting any results. So I moved on to test my protocol. I ran tests using several different protocols, but I found that mine was working just fine, and I was able to visualize both DNA. Finally, I moved on to my primers. So first I diluted them, and then I ran separate PCRs with the same DNA materials, but with different primers. And after running several of those, I came to the conclusion that my primers may be the reason why I'm not getting any results. This is the final PCR that I ran, and you can see that in those empty wells, those were the samples that I ran with my primers. And then the 3 wells that had bands are samples that were run with different primers. So I could conclude that my PCR was working. So while I wasn't able to procure definitive results on the health of the trees in time for this presentation, running all of these different trials allowed me to narrow down the possibilities of what could be the issue. Like I said before, it may be the primers. And another possibility is that none of the branches that I tested have the scorch. My positive control was a tree that had tested positive previously. But like I said before, decline is very gradual, and symptoms don't usually show up until late summer and early fall. So it's hard to tell which branches exactly have the have the bacteria. So while I don't have definitive results, I do have a clear idea of what can be done in the future to diagnose these trees. And in the meantime, I decided to examine the relationships between the leaf traits that I had collected, because that would help me work towards diagnosing the trees and also just observing the health in general. My first question then was, do the cardinal directions affect leaf traits? So I ran some box plots in R, and I found that branch direction didn't really seem to have much of an effect on the leaf traits. You can see here that all the boxes are lined up pretty nicely, and the p-value is not significant. So then I moved on to another question. Does age affect leaf traits? For 2 of my collections, all of the trees were the same age. So for my result, I can't say for sure that it's just age that's affecting what I'm testing. But if you look here, it seems like there's a very strong positive correlation between percent nitrogen and age, and there's a significant p-value. So just looking at this information, you would think that there is a relationship between percent nitrogen and age. But when you add location into that analysis, you can see that there might be other factors that are affecting it. All of the trees that were 12 years old were in the tree breeding nursery, and they all had lower levels of percent nitrogen. Again, we can't say for sure that that's just because of the age, because it may be that the tree breeding nursery didn't have as good soil quality as the rest of the locations that I was testing. And you can also see that the trees from the plants from China collection all had a much higher average percent nitrogen than all of the other trees, including the 23-year-old. That one seemed to have a higher percent nitrogen than even the 27-year-olds from the other collections. Another factor to consider is that the trees may not be healthy. So I went out last week to go study my trees to see if I could pick up any symptoms, and I found that the tree with the lowest percent foliar nitrogen was also exhibiting symptoms of the scorch. You can see the banding up there. So while I can't say that age is affecting nitrogen or location is affecting nitrogen, this information could be used in future studies. For percent carbon, again, there's a significant relationship. It's negative this time. And just from looking at the first age group, you would think that location doesn't really have much of an effect. But if you take a closer look at the red and purple dots, those are the oak collection and tree dissection trees, those all had very, very similar readings for average percent carbon. So again, mixed results. We can't say definitively whether or not age and location have an effect. For leaf area, again, significant p-value. And actually 3 out of the 4 collections seem to have very similar leaf areas. But if you take a look at the 2 red dots up there and the 1 red dot in the 27-year-old age group, you can see that there's a pretty stark difference there. The way I interpreted these results was that that one tree may be less healthy than its other counterparts in the same collection. And then for moisture content, there was a significant positive correlation. And again, you can see that there's a difference between the 23-year-olds and the 27-year-old in that collection. Again, that might be because of health, it might be because of age, we can't know for sure. In conclusion, to diagnose these trees in the future, I wanted to use a definitive positive control. And based on the results I got from comparing the leaf traits, while age seems to have a significant effect on the leaf traits examined, future research would have to incorporate important factors like location and health. This project was funded and supported by the Morton Arboretum Center for Tree Science and Morton Solved. Special thanks to my mentors, Chuck Hannon, Lane Scher, Stephanie Adams, and Chai Chang Hua, as well as to the many people and facilities on their list for advising and assisting me throughout this process. Thank you.
So once a tree is infected, is it perpetually infected, or is it just with that year's leaves, and so next year it's going to be fine until it gets, experiences the vector again?
Yeah, it's a chronic and eventually fatal disease, so they usually leaf out pretty normally the first year, but then as years go on, the symptoms just become progressively worse, and the tree will die.
How does the tree get this again? How is this pathogen happening?
So it's usually vectored by insects that feed on the xylem fluid. It can also happen when an injury occurs to the tree, and it basically causes just these air bubbles to form in the xylem, and that will essentially strangle the tree.
So is there any kind of treatment or remediation? I mean, how do people deal with this?
There's no cure. There are certain steps you can take to promote the health of the tree for as long as possible. Like you can prune away the dead branches. I think there's some culturing that you can do with it, but eventually the tree will die. So efforts are, I think, mostly focused on prevention.
What other efforts in controlling the insect that is carrying this disease? Like, is this insect invasive, and are there protocols for controlling its population?
I haven't seen too many protocols for that, at least in the research that I looked at, but I'm sure that there are some studies that are working towards that.
So is this disease native to the United States? Or was it brought in from somewhere else?
I don't think it's brought in from anywhere else, but I know that there have been occurrences in the East Coast, the Midwest, and the South.
I think it's moving.
It's quite widespread.
I would actually like to make a comment as faced with an unusual number of technical challenges. So I'm all about technology, and I was pushing technology on my students here. And the first plan was to do a drone project, and kind of a couple of weeks into this, it just wasn't really working, and it kind of turns out it was maybe the tablet. I didn't buy a good enough tablet, so the drone project kind of died, and then she's in PCR hell for several weeks, and then the Elementar to like measure and she had to chase down 3 different facilities before she could get all of her. So she worked very, very hard